proteins into enzymes that really allow for life as we know it to develop. Then we have the defensive proteins. Defensive proteins are antibodies. Antibodies that are generated by the B lymphocytes in our bloodstream, and antibodies that recognize viruses, that recognize bacteria, and also that recognize what we call foreign proteins that get introduced into your system. Then we have storage proteins. Examples of storage proteins are milk, for example, or ovalbumin, the egg white in eggs. And what storage proteins do is it's just simply a protein that's composed of amino acids that gets broken down. So these amino acids can be used for the embryo, for the developing embryo. And what is the process that breaks down ovalbumin? Remember, proteins are made by dehydration synthesis. So in the breakdown of ovalbumin, what are you using? Hydrolysis, right? The reverse reaction. Okay, then we have transport proteins. Transport proteins are responsible for carrying cellular contents from one point to another, or, for example, hemoglobin is not working within the cell so much, but it's actually in the blood. It actually carries, transports oxygen from the lungs and other parts of the body. We also have, like I said, transport proteins that work within the cell. We have all kinds of transport proteins embedded in our cell membranes that carry things in and out of the cell. We also have hormonal proteins. Hormonal proteins, or they're called peptide hormones, are used as signals for target cells to change the behavior of target cells. We have receptor proteins. Receptor proteins are proteins situated on the surface of a cell, and they respond to certain types of chemical stimuli. And a wonderful example of receptor proteins are the signaling molecules that are released, or neurotransmitters are examples of signaling molecules. Neurotransmitters are signaling molecules that will be released from a nerve cell. And then there are receptor proteins on an adjacent nerve cell that can bind to these neurotransmitters, and the receptor proteins cause the cell to change its behavior. All right. We also have in our cells contractile and motor proteins. These are proteins that are involved in cell movement, and that would include, for example, structures such as cilia and flagella that are associated with motor proteins that cause these structures to actually move. One of the most important motor proteins that you already know about would be actin and myosin. These are the motor proteins that you find in your skeletal muscle tissue, for example. And then finally, we have the structural proteins. Structural proteins are proteins that are generated by cells in order to support tissues, or support proteins that are used for other types of functions. And a very good example of that is keratin. Keratin is the protein that you find in hair, horns, feathers. But keratin is also the primary component of our skin. The skin that you're sitting in is composed of keratin. Okay? And we'll go over that later. The cells, all of your cells produce all kinds of what we call matrix proteins. Your cells don't just sit by themselves. They secrete proteins to give themselves support. That's called the extracellular matrix. The extracellular matrix contains proteins secreted by the cell to help support the cell. And a very good example of that is collagen. And I know you've heard of collagen. Okay. So these are the 8 primary categories of protein. And like I said, one of the most important to have evolved that has allowed for life to develop as we know are the enzymes, simply because the enzymes will accelerate biochemical reactions that allow for a cell to live and function. If you didn't have enzymes, the biochemical reactions that occur within our cells would take far too long. Okay? Can I repeat anything or explain anything further? Are we clear? Question? The question is receptor proteins. What do they respond to? In this particular example you're looking at on the slide, this would be a nerve cell, and it's releasing what are called neurotransmitters. And for neurotransmitters to work, what happens is an adjacent cell has receptors on its surface that bind to these neurotransmitters. When the neurotransmitters bind to their cognate receptor, it changes the behavior of the recipient cell. Okay, question?
You said there were two important proteins and all of my device that enzymes that we do.
Well, that would be it. Enzymes. The question was, what was the other important protein for life as we know it, and it is enzymes. They're the most important. Okay. So when we were talking about functional groups, if you recall, we talked about amino groups and we talked about carboxyl groups, and the example we used for both was an amino acid. And so we're going to revisit the structure of the amino acid. An amino acid has what we call an alpha carbon. Arranged around the alpha carbon, you have, like we learned earlier, an amino group. You have a carboxyl group. The amino group, if you recall, can function as a base. The carboxyl group can function as an acid. The alpha carbon always is also associated with a hydrogen, covalent bond hydrogen, and then to an R group. We have 20 different amino acids. They differ by their R groups. It's the R group that distinguishes one amino acid from the other. And it's the R group that really conveys the chemical and physical properties of each amino acid. And here are the 20 different amino acids. You are not going to memorize any structures. Do not do that to yourself. There's no reason. You are not going to memorize any structures. The only thing I would ask is that if you look at the R groups, and that's what you're looking at here, this is the common structure of all amino acids. It's shown here in the light lavender. But the R groups are shown in this case in yellow, and these would be the nonpolar or hydrophobic R groups. And what distinguishes these nonpolar or hydrophobic R groups is the inability to form hydrogen bonds or interact with water. You'll see that there's a lot of methyl groups or ring structures or just carbon chains, carbon structures that are not amenable to forming hydrogen bonds. Therefore, they're considered hydrophobic. So I would ask, the only thing I'm going to ask of you is to be able to look at a structure like this or this or this and tell me, can it form a hydrogen bond? And we already have that first principle down pat, right? The hydrogen bond has to be a hydrogen shared between an electronegative atom, and those electronegative atoms are oxygen and nitrogen, right? And so can this R group form a hydrogen bond with anything? Absolutely not. Absolutely not. So that's what you need to know. That's what you need to know. You're not going to memorize structures, but you're certainly going to understand the types of chemical reactivity that the R groups can undergo. Okay? All right, so now we're looking at not the hydrophobic, but now we're going to look at some hydrophilic R groups. And they're hydrophilic because, again, they can form hydrogen bonds with the surrounding aqueous environment. The exception is cysteine. Cysteine is considered hydrophilic, although it doesn't really form hydrogen bonds because you're looking at a sulfhydryl shown here. It's still considered hydrophilic. The rest of them, serine, threonine, tyrosine, asparagine, and glutamine, all have the capacity to form hydrogen bonds with the water in their surrounding aqueous environment. So you can determine the hydrophilic amino acids. You can certainly say, well, this can form a hydrogen bond, whereas this one really can't. Really can't. Okay. Tryptophan, it's interesting. Tryptophan, I have that stopped because you have a nitrogen with a hydrogen. This could form an amino acid. And actually, oftentimes what you'll find is tryptophan will actually be the amino acid that it functions as a boundary between an aqueous and a hydrophobic environment. Okay? So tryptophan is rather interesting because by virtue of this nitrogen and hydrogen, tryptophan, due to its fused ring structures, is overall hydrophobic. But with this nitrogen and hydrogen, there's a certain amount of hydrogen bonding it can be involved in. Therefore, you frequently will find tryptophan on the boundary of an aqueous and hydrophobic environment. Okay? Does that make sense to you? It's logical, right? Okay. Then we're going to come down to another group of hydrophilic amino acids, and these would be the charged amino acids. That would include aspartic acid, glutamic acid, also positively charged amino acids, lysine, arginine, and histidine. These are, the acidic are negatively charged, the basic are positively charged. And what I want to point out, looking at the structures and properties of these different R groups, and all of them, like I said, all of them have a common backbone, but it's the R group that will distinguish the properties of the amino acids and their structures and behaviors. So what I want to point out is that by virtue of their charged nature, the amino acids, aspartic acid, glutamic acid, lysine, arginine, and histidine, actually can be involved in ionic bonds. They can be involved in ionic bond formation, as well as hydrogen bond formation. These hydrophilic amino acids can be involved in, of course, hydrogen bond formation. The cysteine gets involved in what you would call sulfhydryl. So you have what you call a sulfur bond that results. It's a disulfide bond, I should say. And then the hydrophobic amino acids that you're looking at here with their methyl groups or their fused rings, these amino acids are usually what you'll find on the interior of a protein, away from the aqueous environment. And we'll go into that in a little bit more detail shortly here. Does anybody have a question, or can I repeat anything? Okay. So let's talk a little bit about peptide bond formation. Peptide bonds occur on the ribosome in the cells. Peptide bonds happen on the ribosome. It's on the ribosome that peptides are formed. And so what happens is dehydration synthesis, so you'll have a growing peptide chain, or where you have amino acids linked together, and an incoming amino acid is going to be linked to the existing peptide chain through dehydration synthesis. What we call this, the carboxyl group on this end of the peptide is going to donate a hydroxyl. This nitrogen, the amino group of the incoming amino acid, is going to donate a hydrogen to form a water molecule. And when these two groups are split off, a covalent bond is formed between this carbon and this nitrogen, and that is the peptide bond. We're going to go over this in a lot more detail, a lot more molecular detail when we talk more about protein synthesis later on. But all I want you to understand is that you have a growing peptide chain that the growing peptide chain will have an amino terminus that will be defined by its amino group, and it's going to have a carboxyl end or a carboxy terminus. And this is actually set up the way that proteins are formed, where you have a peptide bond formed between a carboxyl group and an amino group, so that you always end up with the amino group. The amino terminus would be the first amino acid that forms the chain. The carboxy terminus is going to be established by the final or the terminal amino acid in the polypeptide chain. So that's how you get the orientation of proteins with an amino end and a carboxy end. The amino end again is called the amino or N terminus. The carboxyl end is called the C terminus or carboxy terminus. And again, that peptide bond, the way it forms, helps to establish the orientation of the N terminus and the C terminus. Does anybody have a question? Can I repeat anything? Question? Okay. All right. So, as I was indicating earlier, let's take a look. It's these R groups. The R groups and their physical properties, whether they're hydrophilic, hydrophobic, or charged, and they can get involved in ionic bonds or hydrogen bonds. It's the physical properties of the R groups that will determine how this polypeptide chain that is forming, how it's going to fold onto itself to form what we call a mature protein. So as a polypeptide chain is being formed, as it's being formed, depending on its amino acid sequence and the properties of the R groups on the amino acids, that's going to determine how that polypeptide is going to start folding upon itself and forming a final structure. And it's when a polypeptide reaches its mature folded state and becomes functional, that's when it becomes a protein. So you could call an immature protein a polypeptide. A polypeptide is a series of amino acids being linked together, and determining the R groups are what's determining how that series of amino acids is going to force the polypeptide to fold and take on an eventually a functional shape. And when that polypeptide takes on its mature functional shape, that's when it becomes a protein. That's when it becomes a protein, when a polypeptide takes on its functional shape. Okay? And what determines the functional shape? The R groups of the amino acid sequence. All right. And so what you're looking at here are examples of, first, a ribbon model or a space filling model of a well-known protein called lysozyme. This is actually a protein that you find in your tears and your saliva, and it binds to bacteria and prevents infection. Did you know you had that? Not very many people have bacterial infections of the eye, and one of the biggest reasons is lysozyme. And you're looking at it here, and it has, by virtue of its folding patterns, it will have a very specific functional structure, including this groove that you see here, which is where it's going to bind to bacteria and prevent the bacteria from infecting your tissues. Okay? What I also want to point out is that if you look at your amino acid R groups, it would be these hydrophobic amino acids. These hydrophobic amino acids that you see here, that's what's going to be on the interior of the protein, away from the aqueous environment. So the hydrophobic amino acids will bury themselves away from the aqueous environment. They'll bury themselves in these helical structures deep inside, whereas the hydrophilic amino acids or the charged amino acids, those are the amino acids that you're going to find on the surface of the protein because they're comfortable in interacting with the aqueous environment. Does that make sense to everybody? Yeah, it's very logical. Okay? All right. And by virtue, like I said, by virtue of these R groups, the R groups will help to determine just how the protein folds to take on its final structure. And when when the polypeptide takes on its final structure, that's when it becomes a functional protein. An example are your antibodies. Antibodies take on a very, very distinctive structure that allow for them to bind to viruses or bacteria that are present in your bloodstream. And what I want you to notice is by virtue of the folding, that's what allows for proteins to interact together. They will actually have a folding pattern that will drive their function. And for example, with an antibody protein, that antibody protein is going to have a very specific structure that will allow for it to interact directly with a protein that you would find on a flu virus or a bacteria. That's what allows the antibody to specifically recognize a flu virus or bacteria and then dispatch the virus or bacteria before it can do serious damage to your cells. Okay? All right. Now I'm going to talk about the levels of protein structure. Proteins have 4 levels of structure. The first or primary level of structure is the amino acid sequence itself. So you're looking here at an amino acid sequence of protein called transthyretin. It has an amino terminus and a carboxy terminus, and again, that's established by virtue of the way that the peptide bonds form between the component amino acids. So the amino acid sequence of a protein, the amino acid sequence is actually the primary structure. The amino acid sequence is the primary structure. Then we have what is called secondary structure. Secondary structure occurs through hydrogen bonds that form between let's go back. Secondary structure forms by hydrogen bonding between the nitrogens in the polypeptide backbone and the carbonyls. So nitrogen with its hydrogen will be able to share this hydrogen with the oxygen of a carbonyl and set up a hydrogen bond. And it's these hydrogen bonds that form between the oxygen of a carbonyl and the nitrogens on the peptide backbone. This is what establishes what we call secondary structure. And secondary structure, because of these hydrogen bonds that are forming, secondary structure will actually form what we call an alpha helix, where the polypeptide will actually form a helical structure, or the polypeptide can, using hydrogen bonds, form what we call a beta pleated sheet. A beta pleated sheet. And just to give you an idea of what that would look like in a mature protein structure, let's take a look. Here's a good one. Look at this. What we're looking at here are some alpha helices. These are alpha helices. And then buried within lysozyme, you also have some beta pleated sheets, shown here. What you also have, there are segments of a protein that will not take on that form of secondary structure. They won't form beta pleated sheets or alpha helices. They will form what we call a coiled coil. And you're looking at that right here. These are oftentimes called linker regions. Oftentimes called linker regions, where they link areas of beta pleated sheet the areas of alpha helices. And let's take a look again at this example. So here you're looking at an alpha helix. Here you're looking at a beta pleated sheet. And here you're looking at the coil region or linker region. This is a segment of a polypeptide chain that doesn't form either alpha helix or beta pleated sheet. What I also want to point out too is that the beta pleated sheet is interesting in that the way the protein will fold, or the polypeptide will fold, is it will fold in one direction and it will establish hydrogen bonds with an adjacent segment of the polypeptide that is going in an opposite direction. So beta pleated sheets are actually what we would call anti-parallel structures. They're anti-parallel, and that anti-parallel structure, again, is established through hydrogen bonds that form between the, go back, I'm sorry I keep going back, but I just want to make sure everybody understands. Hydrogen bonds that form between the nitrogen and associated hydrogen and an adjacent carbonyl, the oxygen on that carbonyl. Okay. And that's what you get with beta pleated sheets. You'll find them in what we call anti-parallel arrangements within the folded structure of a protein. Can I explain anything else again? Are we okay? Okay. All right. Question? Question?
Call the connecting part between the helix and the pleated sheet.
Okay, this connecting part, it's called a linker region,
Okay.
and it's also sometimes called a coiled coil.
Okay.
Doesn't really form a helix or a beta pleated sheet, but it's critical for connecting these segments together. Okay?